So now let's welcome Dr. Melamed, Dr. Doron Melamed from Technion, from Israel, who is going to talk about the effects of aging on B lymphocytes and the potential for depleting of the bone marrow of the aged cells.
Okay. I'd like to thank Aubrey and SENS Foundation for inviting me to here. I'm very happy being here. It's my first time in this type of meeting because I'm a developmental immunologist. And my lab is focusing on B cells and development of B cells. And we have recently got into the area of aging in the B cells, which is very fascinating. And what I would like to show you today is a very simple approach that we are applying, and we obtain rejuvenation of the B-lineage. Now, my talk will be based on many immunological models and many mutants. And I am therefore hoping that the comprehensive introduction that you have heard earlier would support the ability to comprehend the complex data here. So anyway, when we look at the immune system in age, along the age, along life, it's basically what you see in any other physiological system. And you can do any kind of measurement either by production or function, either in the whole body, organ, or the specific system. You can see that in childhood there is a rapid increase in that, and then you get into an early adult stage where you get to a plateau. And later in adult life, you'll start seeing decline in the function, and in aging, it goes further down. Now, we've heard a lot in this meeting and in the last few years about several approaches, medical improvements, either environmental or drugs, where we have basically been able to shift this decline a little bit and to increase the life expectancy by a few years. But it is most important to note that this decline or the shape of this curve stays the same thing and does not really change. Now, when we approach this question, we asked for more dramatical changes, is whether or not we can change the shape of this graph by elevating or by modifying this decrease, and maybe to shift it up or to make it a little bit less sharp, or even to maintain this the function as it is in the adult early lifespan. So in the immune system, as we heard earlier, is aged, and this applies in any compartmental. So we see defects in B-lineage, in the development and function. We have just heard about T-cell defects similarly, and defects in the innate immunity. These defects lead to increased susceptibility, increased morbidity in aging, and you see here that around the age of 40 years old, you start to see more infectious diseases such as influenza, pneumonia. And it's important to note that these diseases are in addition to other age-related diseases that we have heard about it, which are mostly neurodegenerative metabolic diseases that are age-related and not immune-related. Though there are many immunologists you would hear that will say that everything is immune or autoimmune unless otherwise proven. Now, If you want to combat this increased susceptibility, this can be achieved in young, of course, by vaccination. But as you see here, and we've heard earlier, the elderly population is poorly responsive to vaccination. In this study, over 1,800 people were vaccinated that are aged 60 years or older, that were vaccinated against influenza. And you see that about 50 to 60% of the people only develop protective antibody. We could easily recapitulate that in a mouse model, in mice in the lab, where you immunize mice with an NP. This is an antigen, specific B-cell antigen. And you see that young mice develop nice antibody amount of antibodies, whereas old mice fail to do so. So what is it in the B cells that is happening in aging? So if you look at this cartoon, this shows the stages in B cell development. And the development occurs mainly in the bone marrow, where B cells progress from one stage to another, where they go through immunoglobulin rearrangement to assemble and express the B cell antigen receptors, the heavy and the light chain, on the surface. Once these cells express it on the surface, they are then subjected to a negative selection where self-reactivity is eliminated. And those that do not bind a self-antigen, then they are subjected to what we referred as positive selection and maturation of the cells from the bone marrow. They are enabled to migrate to the periphery to complete their maturation. In the periphery, they reside and they form the huge diverse repertoire that is capable to respond to and recognize any given pathogen. So now what happens in aging? If you look at the main two compartments of B cells in life, in an individual, in the bone marrow and in the periphery, in the young situation, you see that the bone marrow is making a lot of B cells, and constantly these B cells migrate to the periphery to replace the naive B cells, which are relatively short-lived. And this is constantly occurring, and this constant replenishment occurs in the young situation. However, while facing pathogens, slowly you accumulate more and more memory B cells. These are shown in red. Memory B cells are long-lived, and they have and they by themselves can go some proliferation. Now, because of homeostatic forces, the size of the B cell pool in the periphery doesn't change with age. So the increased frequency of the memory cells become to be on the expense of the naive B cells that in the periphery. In parallel, the bone marrow production of B cells declines. This results in a relatively restricted repertoire of B cells in the periphery. And because of the restricted repertoire, now there is no recognition of new or evolving pathogen in aging. And this is similar to what we have heard in T cells in the previous talk. If you want to visualize these changes, we can simply analyze B cells production in the bone marrow. So this is an analysis of bone marrow in young and old. You see that by staining of B220, B220 is a B cell marker, IgM is the B cell receptor. Just by this simple staining, you can distinguish three major populations. The pro and pre-B, these are the earlier precursors, and the immature cells. These two populations are newly generated in the bone marrow, and the circulated cells, which comprised of mature B cells that are circulating through the bone marrow. You see that in the young, the majority of the B cells are belonging to the pro and the pre-compartment and the immature, and few are in the circulated. In adult, you see that this earlier precursor, or the bone marrow-derived precursors, they are not, or their frequency is very low, and most of the cells in the bone marrow are recirculated B cells. Now, what is the cause of these changes? There are two mechanisms that explain this. The most accepted mechanism, the widely accepted mechanism, is a mechanism that based or predicts that aging resulting from defects in B-lymphopoiesis in the bone marrow. According to the process that causes it is that early progenitors, they acquire defects with aging, acquire defects that are mainly irreversible. Now, these defects cause reduced production of B cells in the bone marrow and reduced output to the periphery. In order to maintain the number of B cells in the periphery, there is selection for long-lived peripheral B cells, and the outcome, of course, is limited repertoire and poor responsiveness. The prediction here is that aging is progressive and unstoppable, and of course, it is irreversible. We approach this by bringing another possibility, is that aging is basically the consequence of homeostasis. Basically, if we want to simplify it, is to say that aging is the outcome of demand and supply. Accordingly, we thought that in aging there is accumulation of long-lived B cells in the periphery. Now, this accumulation, these B cells, now suppressed by some kind of a mechanism that is yet to be discovered, they suppress the process of B-lymphopoiesis in the bone marrow. As a consequence, there is a reduced output of B cells from the bone marrow, resulting with the same thing, limited repertoire and poor responsiveness. However, in contrast to the previous model, this model, because it is based on homeostasis, predicts that if we are able to control or to manipulate homeostasis, then we can reverse aging. How do we prove it? The best way for us to approach this is to use a mouse model that has impaired B cell maturation. Now, if it has impaired B cell maturation, this mouse will have chronic B cell deficiency. So the number of B cells in the periphery will be chronically reduced and not achieving the number that are supposed to be in a normal mouse. Now, if aging resulting from irreversible defects, then the prediction is that this mouse will gradually lose all the B cells and become in age to be a B cell-less mouse. However, if B cells is a matter of homeostasis, then the prediction would be that these mice, when they age, their phenotype will be just as a young mouse. So a young-like phenotype will maintain through aging. We have used a mouse that is deficient of CD19. CD19 is a co-receptor that is expressed on the surface of the B cell, and it balances B cell receptor signaling. CD19 is a positive regulator. This is just some notes. It's a positive regulator. It required for maximal PI3 kinase activation and calcium mobilization, and it recruits other signaling molecule like VAV and LIN upon BCR ligation. Mice that are deficient of CD19, they have relatively normal B cell development in the bone marrow, and their peripheral B cells are short-lived. They have about 50% of the B cell numbers, and the B cells are hyper-responsive to stimulations. In several papers that we have published in the past, we have shown that these B cells have inappropriate tonic B cell receptor signals, and therefore they fail positive selection and maturation, and consequentially CD19 deficient B cells have chronic B cell deficiency from birth in the periphery. When we look at these mice, and we look first at the peripheral B cell numbers, we can see that, first of all, if we look at the wild type and compare young and old, we see that the number of B cells in the periphery or in the spleen are basically equivalent, and this is what the literature says. In the CD19 deficient mice, first of all, you see that they have reduced numbers. It's about 50, 60% numbers. But this stays also in the age. So old mice do not lose. They have the same number of B cells as young mice. We have also recapitulated all the results that obtained in CD19 in another mouse model, the CD74 deficient mice. CD74 is a molecular MHC chaperone that mice that are deficient in that exhibit a very similar phenotype as CD19 deficient. I am not going to get into the details, but they also have a reduced number of B cells in the periphery, chronic deficiency, and everything that we have done with CD19, we have recapitulated also in this model. So you see first that this mice model, they don't have reduced B cell numbers in the periphery. Now, if you look at the bone marrow, okay, this is again a wild-type mouse. You see young in the upper and old in the lower. You see the phenotype of the age mouse, reduced frequencies of the earlier precursors. But if you look at the CD19 deficient, you see that the number of B cells are significantly enhanced in the bone marrow. So the bone marrow of the old CD19 doesn't really age. And this is the case also in CD74 deficient mice. Just compare this to the wild type setup. Now, because these mice have a developmental impairment, though in the late ages, we have recapitulated the whole thing in a third model, which is mice that are deficient of BAF signaling. BAF is a survival factor for B cells, and it's for mature B cells, but it's dispensable for development. So mice that are deficient of BAF signaling have reduced mature B cell numbers because the B cells fail to survive. Okay, so these are BAF receptor deficient mice, so they don't have this signal. You see, these are wild-type mice. Again, there are no the reduced numbers of the early precursors in the bone marrow, whereas the BAF receptor deficient mice, you see that they don't age. The old and the young look just as similar. If you look now at the stem cell population, you see that stem cells develop from long-term repopulating hematopoietic stem cells. They progress into multipotent primitive progenitor and then to common lymphoid progenitor before they go to the B-lineage. It is known that in aging, this progression is inhibited in a way and resulting in accumulation of these cells, of the long-term hematopoietic cells in the bone marrow, and reduced numbers of MPPs and CLPs. This is the situation in the old, but you see that in the mouse that has chronic B-cell deficiency in the periphery, this phenotype, it does not show up. You see equivalent numbers of all these three populations in the bone marrow, indicating for a continuously development, continuously differentiation as well in the old situation. So now what happened in the periphery? In the periphery now, we have used another mouse model. This is an immunoglobulin transgenic mouse, which is used as a repertoire reporting model. What is the immunoglobulin transgenic mouse? This is a 383. In this mouse, all the B cells express the same receptor on the surface. This is because of efficient allelic exclusion. We call it a monoclonal mouse. So you see, using an antibody that is specific for the idiotype of the receptor, you can identify and show that all the B cells express the same specificity. This is in the periphery. However, as reported by a group, by John Cambert's group from Denver, Colorado, in aged mice, there is a selection process which allows cells, variant cells that express an endogenous receptor to accumulate in the periphery. And in fact, in old mouse, 50% of the cells express endogenous receptors. This transition occurs only with aging. Now, if you cross this mouse to CD19 deficient mice that bear chronic B-cell deficiency, you see that in a young mouse and in old mouse, most of the B cells in the periphery retain expression of the transgenic receptor. So basically, the old phenotype that is occurring in the old mouse does not occur here if your mice are CD19 deficient. So the conclusion for this is that aging in the B-lineage is mediated by cellular homeostasis, and then chronic deficiency from birth prevents age-related alterations in the B-lineage. Now come the other question. Can we reverse aging in the B-lineage? Sorry. For this, we adopted another mouse model, a model which is utilizing the Kree-Lox system, which allows an inducible ablation in vivo of a specific locus. Here we have generated a mouse where the BAF receptor gene is fluxed by two LOX P sites. Now, these mice are transgenic for an MX Cree, which by injecting the inflammatory mediator poly-IC, you generate interferon, and this interferon binds the promoter, activates Cree, and you start producing the Cree recombinase. The Cree recombinase then cut these two LOX P sites and delete the locus between the two. So you can generate, and how does it look? So you see that this is a heterozygous situation, this is the homozygous situation, this is the number of B cells that are found in the peripheral blood, this is just young mice. You see that when you introduce interferon, you introduce the poly-IC, the number of B cells drop by about 50%, owing to the lack of survival of the cells. They now don't get the BAF signal. And you start see many newly generated cells in the peripheral blood and in the spleen, as you see by using a marker that is specific for newly generated cells in the bone marrow. Okay. Don't worry. So now, if you do that in an old mouse, and you apply this depletion system, you see that this mouse have regenerated the whole B-lineage in the bone marrow. This is an old BAF mouse treated with this. You can quantify now the number of B cells and look at the pro-B, pre-B, and immature. You see that the vast majority of the B cells in the bone marrow of this old mice belong to the early lineage. This is relative to the old one. So aging in the B-lineage can be reversed. Now, can we do that in wild-type mice? Here, the hypothesis would be that if we take the old situation, we modify this homeostasis by depletion of the B cells. Now, this would revive B-lymphopoiesis in the bone marrow and rejuvenate the peripheral repertoire. Does this work? First, this is the antibody administration. You see, this is the number of B cells determined by CD19 expression. This is the number of B cells in the peripheral blood. After antibody depletion, you see that they are dropped dramatically. And then you can follow the kinetics of B cell return. You see that the B cells are depleted nicely, and then they slowly return. You see that in the young mouse, they are rapidly reconstituted. In the old mouse, they are slowly reconstituted. I will come to this in a minute, but let's go first to see what is going on in the CD19. Remember, CD19 doesn't age, and you see that young and old mice equally reconstituted their peripheral compartment after depletion. Now let's go back to here. Why is there a difference between young and old mouse, between the rate of B cell return between young and old mouse? We attributed that to the lower frequency of precursors that are committed to the B-lineage in the old situation. Now, how can we prove this? We can prove this by applying multiple rounds of depletion. So if you take old mice and you deplete the B cells, the first round of B cell depletion, the B cells comes very slowly. But afterwards, if you apply second round of depletion, the mice reconstitute faster. And the third depletion, the reconstitution rate is not different than what you see in young mice. If you look at the bone marrow of the mice that have passed through 3 cycles of depletions, you see these are wild-type mice. You see that the B cells in the bone marrow is completely restored. Now, what about the peripheral compartment? Here again, going back to the model, the 383 transgenic system, you see that after depletion, the bone marrow start making new B cells, and the phenotype that is generated by this bone marrow is a young-like phenotype relative to the old, that 50% of the cells are expressing endogenous receptors. Finally, does this change the response? So if we rejuvenate the peripheral compartment, do we get now a new response, an improved response? The answer is yes, but not as much. So if you see this is an anti-NP response, you see this is the young response, you see this is the old, this is antibody response, anti-NP, anti-IgG1. You see this is the old mice response, and this is the response that is generated by the old mice that have been treated for B-cell depletion. So you see that there is a dramatic improved response, but it's still not as the one in the young situation, owing to the fact that these mice, have also old T-cell compartment that yet or need to be rejuvenated as well, hopefully, to get this thing. So this conclusion brings me to say that B-cell depletion rejuvenates the peripheral repertoire, and that the rejuvenated repertoire mounts an improved antibody response to new antigenic challenge. And then going back to my first slide, I think we were able to show that this slope can be shifted by B-cell depletion to reactivate B-lymphopoiesis in the bone marrow. And in a situation where you have chronic B-cell deficiency, you can basically maintain B-cell production in an old mouse throughout life and through aging. And I would finally like to acknowledge the people who did the work. This is people in my group that contributed, and these are my collaborators in Europe and in USA. Thank you very much for your attention.
So, very interesting talk. We expect a lot of questions, but we can take only maybe 3. So, Yanko?
So your data suggests that there is a feedback of the mature memory compartment onto the bone marrow. Any more insights that you can share with us along those lines? How does it happen? What is responsible? And then the second question is, when you do your serial depletion and you get your improvement in the function, how long lasting is that effect?
For the first question, my answer is I don't know. These are experiments that are ongoing. We have few other models where we assess both the effect of soluble factors and some cellular. There are some cells that we think may contribute to this phenotype, and we are assessing it now using variable models and experiment both in vivo and in vitro.
Because I guess memory B cells actually like to be in the bone marrow.
True. Yeah. That's true. For your other question, how long it lasts, I can tell. We need to do this. We need to perform these kinetic experiments. I think our first attempt was to bring this as a proof of concept that this can be done and can be achieved, and how can we manipulate that, and how long this lasts, and what is the efficacy? I think that it is yet to be pursued either by lab work and by company or by any other means.
So next question.
Yes, as you know, aging is associated with an increase in autoimmunity. Do you think that depleting the B-cell pool will work improving autoimmunity, that is reducing the incidence of autoimmunity in the old animals?
I can tell you that one approach for treat autoimmunity, or at least some autoimmune diseases, is by using B-cell depletion approach. This is done for rheumatoid arthritis. This is done for lupus. There are many other autoimmune diseases that are now on clinical trials, like multiple sclerosis. I mean, even so B-cell depletion therapy is used in many autoimmune diseases, either as approved or in clinical trials. And at least for the autoimmune diseases, it shows in many of the studies, it shows much beneficial, much benefits.
Thank you. There was a question over there, and we need a microphone here.
Two questions. One, the murine immune system has...
Can you speak louder a little bit?
The murine immune system has many similarities to human, but also many differences.
Two.
How applicable is your model to human B-cell condition? And second...
I apologize that I can't hear
Just
the
a little
question.
bit louder.
The murine immune system has many similarities to human, but also many differences. How applicable is your model to the human B-cell condition? And second, when you deplete the peripheral B cells, you lose the benefit of the memory of those B cells, while you gain the benefit of the naive B cells.
Okay, you kind of touched the two important points in all our approach. The first one, how it is applied to human. We are now testing this in a clinical trial that is running in our institute in collaboration with the hematology department in Rambam Medical Center in Haifa. So we don't know how this applied. This is now ongoing study. As for your second question, it is true. By depleting the memory B cells, you're kind of getting out your memory or your historical or your immune history. But what we wanted to show here is a proof of concept. But as you saw here, you don't need to deplete all the B cells. Because if you remember, the BAF deficient mice, they have 50% B cells. So it might be that 50%, 30%, 20% depletion would also be sufficient. So you don't need to wipe out all your memory. You might be need only to wipe 20%, 30%, which will be sufficient to reactivate the whole B-lineage again. I mean, I don't know. This yet to be tested in an experiment, in vivo and in vitro, in humans, in mice, etc.
Thank you, Doron.